All right. Looks like most people tuned in already. So I think we're going to start. So welcome everyone to this IBM seminar. Today we host Lucca Sielaff from the Max Planck Institute for Multidisciplinary Sciences from the group of Marina Bennati. And a little bit about Lucca. Is a young grad student, as I said, at the Max Planck Institute. But before that, he got in touch with EPR during his bachelor and master degrees. It was in chemistry at the University of Freiburg. And today he's working on developing novel experimental methods for EPR spectroscopy, in particular hyperfine spectroscopy. And he's going to tell us about the development of time domain ENDOR, which is a quite exciting topic. So Lucca, the floor is yours.
Okay, so thank you for the kind introduction, and also thank you for the opportunity to share my work here at the IBM seminar. And yes, so today I want to talk a little bit about the work of the past 2 years on time domain ENDOR for molecular distance measurements. And so let's straight dive in. So if we think of distance measurements in the context of magnetic resonance spectroscopy or EPR spectroscopy, we might first think of electron-electron distance measurements that we can perform using the DEER experiment. And however, complementary to this, recently also the distance measurement of electron spins and nuclei, and especially 19F, has become very interesting and popular. Because it works in a distance range that is not easily accessible with double electron-electron resonance. And fluorine is interesting because it has high sensitivity because of its high gamma value, and also because it's rare in biomolecules. And these distance measurements will be the main focus of my talk. However, at the end, I also actually want to have a short look into nuclear distance measurements via EPR. So this will also be interesting. So first, the outline. What do I want to tell you today? So first, I want to basically give a little motivation. Why are we actually interested in these new methods? So for this, we of course have to understand what are the challenges of the current 19F ENDOR distance measurements. And then I want to introduce to you dead time-free time domain ENDOR and pulse dipolar hyperfine spectroscopy, and compare the performance of these methods with the usually performed frequency domain ENDOR, and afterwards also talk about how we can extract distance distributions from these experiments, and also what are the limits of these methods. At the end of the talk, I also want to shortly talk about multi-spin effects in time domain ENDOR and how we can measure nuclear distances with it. Okay, so let's start with the first part. What are the challenges of current measurements? So the whole story of 19F ENDOR distance measurements started in our group with the work of my colleague Andreas in 2020. And he looked at these nitroxide model systems with distances in the range of 7 to around 15 angstrom. And you can see that at these larger distances here, we already are basically reaching the resolution limit of the method. And since this initial work, there has been a lot of different groups that picked up the idea of 19F ENDOR and have done some interesting work, especially with gadolinium. We've also heard a nice talk by Alexi in the last IBM seminar. And however, there's still the challenge of the distance limit of the maximum achievable distance. And this still is around 15 to maybe 20 angstrom, depending on what you are doing. And yeah, the question is, can we overcome this limit? Another challenge that is still present in 19F ENDOR distance measurements is the extraction of reliable distance distributions. This is kind of a standard procedure in DIA spectroscopy, so that we directly extract the distance distribution from our data. However, in 19F ENDOR distance measurements, this is not straightforward, and I will explain in a second why this is the case. So to understand these challenges in a little bit more detail, we have to look how actually ENDOR experiments are measured, or how these experiments are performed. And for this, we will have a look at the easiest spin system that we can think of. So basically one electron spin coupled to one nucleus to one 19F, spin 1/2. And in this case, we get a 4-level energy system with 2 EPR transitions and 2 nuclear spin transitions. And the standard way to measure ENDOR in these cases is to use the classical MIMS ENDOR sequence. And you can separate this sequence into a preparation, a mixing, and a detection block. And the main idea is here that in the preparation block, we basically create a polarization grating across the EPR transitions. And then afterwards, we use a selective RF pulse, which is swept in frequency. And as soon as it hits one of the 2 nuclear transitions, this can be seen on the electron spin polarization, and this can be mapped out on our electron spin echo. So in a way, you can think of this method as a quasi-CW acquisition. So basically, we sweep the frequency and see of this RF pulse and see at which point we are resonant with one of the 2 nuclear spin transitions. And if we do this for this easiest spin system, we get the two nuclear transitions separated by the hyperfine coupling, and this hyperfine coupling is proportional to the inverse cubic distance, so it contains the distance information that we are looking for. Okay, so 2 important things that I want to discuss in terms of this sequence for distance measurements is sensitivity and resolution. So for the sensitivity, it's very important to understand that in MIMS ENDOR spectroscopy, we are kind of limited by the blind spot function. The blind spot function is coming from the fact that in this preparation block, we generate electron-nuclear 2-spin order, and to generate this, we kind of accept that we generate a scaling factor for our ENDOR efficiency, and this is dependent on the hyperfine coupling and on the time delay tau. And for a constant tau value, this means that the sensitivity or ENDOR efficiency basically scales with 1 over the distance to the power of 6. And this, of course, is very drastic. And you can see here, for example, in this plot, that for longer and longer distances, we get smaller and smaller hyperfine couplings. And you see that the sensitivity, because of this blind spot function, is in general decreasing. And of course, you could now think, okay, we just basically use longer tau values to challenge this effect. However, at some point, we are, of course, limited by the phase memory time of our electron spin. Okay. About the resolution in frequency domain ENDOR, it was a little bit kind of a topic that I was looking into at the start of my PhD in this group. And back then we were wondering, okay, what is actually limiting the resolution for these 19F ENDOR distance measurements? Or in other words, what is contributing to this ENDOR line width? And in the ideal case, it would be very nice that our ENDOR line width would only be dependent on the distance distribution of our sample, because in that case, we could analyze our data in a very similar way to what people do in electron-electron distance measurements. However, because we perform ENDOR in a quasi-CW acquisition mode, this means that we basically also see all other nuclear spin interactions and not only the hyperfine coupling. And this, for example, could be unresolved nuclear dipolar couplings of the fluorine nucleus with neighboring protons or the solvent. And additionally, we also have the effect of power broadening. So in general, we have a few effects that contribute to this ENDOR line width, which kind of makes it tough or hard for us to directly extract the distance information. And additionally, it also, of course, limits the maximum achievable distance that we can resolve in ENDOR. So to understand these effects, we started to measure these effects using nitroxide model systems. And this was a work also mainly done by my colleague Annemarie and also Andreas. And here, for example, we looked at the effect of the power broadening first. So power broadening comes from the fact that in frequency domain ENDOR, we basically detect the ENDOR spectrum as a convolution of the ENDOR spectrum with the excitation profile of our applied RF pulse. And this means that if we use short pulses with higher RF power, we have a broader excitation profile, and this leads to line broadening in our detected ENDOR spectrum. And you can nicely see here that for this model compound, that if we increase the RF pulse length, so we use less and less RF power, we can narrow down the line within these spectra. A second major contribution that we investigated was the effect of heteronuclear dipolar coupling. So as I already told you, there's basically nuclear dipolar coupling of this fluorine nucleus with the neighboring protons of the phenyl residue. And we now basically chemically suppressed these couplings by exchanging the protons with deuterons. And if you now look at these ENDOR spectra that we acquired with long RF pulses, you see that again we can gain in resolution because we do not have the strong effect of these heteronuclear dipolar couplings. Okay, so we have now basically seen how sensitivity and resolution in frequency domain ENDOR is limited. And now I want to introduce you to time domain ENDOR and how this can help us to overcome some of these challenges. So the idea for time domain ENDOR has been around for a long time already, since the 1980s. And the main idea behind this pulse sequence that you see here is that instead of using a selective RF pulse to sweep the frequency and basically detect the ENDOR spectrum in a quasi-CW acquisition mode, we now want to perform FID detection. And for this, we replace our selective RF pulse with two non-selective RF pi half pulses. And what happens here is that after our preparation block, we generate electron nuclear two-spin order, and our first RF pulse now generates us nuclear coherences. And these nuclear coherences are now can now evolve freely under the Hamiltonian. And because basically these nuclear coherences still consist of the SZ operator here, we are able to basically detect these nuclear coherences via EPR. So the idea is that we use the second RF pi half pulse to again convert these nuclear coherences back to electron nuclear two-spin order, and in the end to electron coherence, so that we can detect it in the electron spin echo. If we do this, in theory, we should get a nice nuclear free induction decay, and after Fourier transform, we basically end up with the ENDOR spectrum. And this method already has the intrinsic advantage that basically in this case, we are of course not limited anymore by the excitation profile of our RF pulses, so we don't have any power broadening effect in terms of resolution. However, you might now wonder, okay, this method has been around for quite a long time. Why are we not using time domain ENDOR more frequently? Well, one of the reasons why time domain ENDOR is not typically applied is the typical excitation bandwidth of RF pulses. So what I have shown you here is an RF nutation experiment that I performed at our Q-band setup with a quite high 600 watt RF amplifier, so with high power. And you see that the maximum or the shortest pi half RF pulses that we can generate are in the range of 4 to 6 microseconds. And if we now go on and calculate the excitation profile of these RF pulses, we realize that we only cover a frequency range of around 200 kilohertz. And this is, of course, a very limiting factor, because for time domain, the ideal case for us would be that we basically excite all nuclear transitions, or basically the complete ENDOR spectrum with one single pulse. However, hyperfine couplings can be in the range of several megahertz. So for a lot of applications, it's not straightforward to use time domain ENDOR. However, for 19F ENDOR distance measurements, where we are particularly interested in these small hyperfine couplings, it becomes interesting again. And basically, in this case, we should only remember that our excitation profile is basically limiting our distance measurements in that way that we cannot really detect distances that are below a certain threshold because we cannot excite the full ENDOR spectrum. So in this case, at below around 8 angstrom, we have problems to excite the full ENDOR spectrum with our RF pulses. Okay, so this was one of the main reasons why all the spectra that I will show you in this talk were basically acquired at our Q-band setup, which you can see here. The advantages of this are, of course, that Q-band is widespread and also highly accessible to a lot of groups, and which is very important for time domain ENDOR, that we can apply these powerful RF pulses that allow us to excite the full ENDOR spectrum. Kind of a disadvantage in a way is that at Q-band frequency, the nuclear Larmor frequency of fluorine and protons are only separated by around 3 megahertz. And this means that if we use protonated nitroxide moieties, we already get some overlap of proton and fluorine resonances. And to basically avoid these overlap for frequency domain ENDOR, but also for time domain ENDOR in the first place, we first went for deuterated nitroxide moieties, where we can look at the 19F ENDOR spectrum without having any overlap. Okay, so let's look at the first experimental results with time domain. So we started out by looking at the same model compounds that I have already shown you with frequency domain. And you can see that for both cases, protonated phenyl residue and deuterated phenyl residue, we can record some nice nuclear free induction decay until delay times of around 140 microseconds. And if we now perform Fourier transform, we end up with basically the same ENDOR spectrum that you have seen before. The line width is only slightly narrower, and the reason for this is that basically we have already almost successfully suppressed power broadening by using very long RF pulses where the effect is only minor. And one interesting thing that we should remember here is that at the start of this FID, you see there's a dotted line. And these data points were actually recreated using simulations. And the reason is here that the initially proposed time domain ENDOR sequence actually suffers from a dead time issue. And the problem is known from other spectroscopic techniques. So because we cannot move our detection pulse over basically our starting pulse here, our first RF pi half pulse, we basically have a dead time that is roughly 1 RF pulse length, and which means in our case 4 to around 6 microseconds. So this means that we have to recreate these first data points. This is, of course, not an ideal case, and actually already in the first initial paper by Peter Höfer and Mehring, there was a way to circumvent this, and this is to use basically a dead time-free version of this experiment. So the main idea here is now to, instead of only using the 2 RF pi half pulses, we now introduce an additional RF pi pulse and generate a nuclear spin echo. And by detecting the nuclear spin echo, we can now freely move our detection pulse over the start of the FID and detect it dead time free. So if we do this now for this model compound here, you can see that we can nicely recreate these first missing data points. And in this case, we measured at 3 different positions in the APR line. And this, of course, leads to the fact that we get orientation selection in our ENDOR spectra. So we only see different parts of the Pake pattern resolved at the different measurement positions. Okay, you might have noticed already that the sensitivity in time domain ENDOR, or the spectra that we can get out of these nuclear FIDs is really good. And to compare the classical frequency domain experiment with time domain, we now performed basically a comparative study using a very similar model compound with a slightly longer distance between the fluorine and the nitroxide. And the idea was that we basically kept all the microwave parameters constant. So we used the same tau value, and the same temperature, same length of the microwave pulses, and so on. And we only tried to vary the RF part of the sequence, so to make them as comparable as we can. And if we now look at the results here, we see that frequency domain ENDOR with a high concentration of 400 micromolar gives us a nice spectrum with 50 microsecond RF pulses after 20 minutes. However, we also see that already at 50 microsecond RF pulses, we get an effect of power broadening. So if we really want to resolve the ENDOR spectrum with minimal effects of power broadening, we have to go for longer RF pulses. And you see this, of course, reduces our sensitivity in this case. And if we now compare these two spectra with time domain ENDOR, we see that for this high concentration, we obtain an ENDOR spectrum with a very, very high sensitivity. And this high sensitivity now actually allows us to go down in concentration, in this case by a factor of 40 down to 10 and even 2 micromolar. And we are still able to detect this ENDOR spectrum without any effects of power broadening, so with very high resolution. And this increased sensitivity, of course, could be very interesting because for biomolecules, for example, we have intrinsically low concentrations and are forced to have basically high sensitivity. So here it could become very handy to use time domain ENDOR. Okay, however, so sensitivity is already, of course, great, but the resolution of both methods is still comparable if we look at the RF 200 microseconds RF measurement and the time domain ENDOR spectra. And the reason, of course, is that, as I explained to you at the start of the talk, that we still have these additional nuclear interactions like heteronuclear dipolar coupling, and at higher fields, maybe also CSA, that lead to line broadening. And we thought, okay, how is there a way to get rid of these additional line broadening interactions? And we thought, okay, it would be nice to use nuclear spin echoes. So because in a nuclear spin echo, we refocus heteronuclear dipolar coupling and CSA. So maybe we can use this. And this basically led us to the development of a sequence that we call pulse dipolar hyperfine spectroscopy. And the idea in this case now is to keep the RF part fixed in time. So we basically now detect a nuclear spin echo. We don't move any of these RF pulses. And we now introduce an additional microwave pi pulse that is swept during the time interval tau RF. And to explain to you what happens now in this sequence, I want to look into this in a little bit more detail. So if we now basically neglect for a second all the pulses that we used during the preparation and detection part of this sequence, we are basically left with these three main pulses. So this pulse is still kind of part of the detection of the nuclear spin echo. And if I now would basically swap the label of this frequency axis with omega observer and omega pump, you would realize that this is basically very similar to it is the 3-pulse Diaz sequence. And a very similar thing is happening in PDHS. So the first RF pulse generates us nuclear coherences, omega I minus, omega I plus. And these nuclear coherences now evolve for some time t. And then a microwave pulse is applied. And this microwave pulse now induces nuclear coherence transfer, so that spins that have evolved with omega I minus will now evolve with omega I plus and vice versa. Afterwards, the RF pi pulse will flip the sign of these coherences, and they will evolve for another time period tau RF. And if we now plug in all the values for the nuclear spin echo and basically see that, for example, the nuclear offset cancels out, we see that our nuclear spin echo gets modulated with the hyperfine coupling as a function of the time t. And this is, of course, basically the same that we see in double electron resonance, or very similar. And so let's look at this experimentally. So the upper part you have already seen. This is basically frequency domain ENDOR and dead time-free time domain ENDOR. And you see we have comparable line widths. However, if you now look at the time trace with the new pulse dipolar hyperfine spectroscopy, or PDHS, you see that we get modulations that lasts until almost time delays of 360, 370 microseconds. And if we now perform a Fourier transform, we see that the line width in this case is massively reduced. So we get an 8-fold decrease in ENDOR line width, so a massive increase in resolution. And we are now actually able to resolve the principal shape of the Pake pattern in this case, which is really nice. And so the resolution improvement is, of course, very great because we can aim for larger distances. But another important thing is that we can now really say that by refocusing these additional line broadening interactions, we can now access distance distributions in an easier way. And before I will talk how to extract the distance distributions, I will quickly want to say two important things about the setup of these experiments because, yeah, they are kind of new. And so there are two important things that we have to consider when we set up PDHS, and one of them is the choice of the length of your time trace or the choice of t. So this is important because of two things. So of course, the choice of t basically determines the maximum resolution that we can get, but it also affects the choice of our tau value that we should use for the measurement. Because if we again look at this time trace again, then we realize, okay, we want to, for example, to basically have maximum resolution, we want to detect this modulation until full decay. And to do this, we need times of 350 microseconds, which means we need a larger value of tau RF. And we have to apply this twice. So this means that our t, which is the delay between the second microwave pulse and the third pi-half microwave pulse, already gets into the range of milliseconds. And in this case, we really have to consider the electron spin relaxation. So T1 is, of course, effective here, but another important point is that also our phase memory time is dependent on the time delay T. So what you see here is a measurement for this molecule of the phase memory time versus increasing time delays. So we use a stimulated echo and detect the phase memory time. And you see that the phase memory time is decreasing with increasing time delays of T. And this means, of course, that if we use a longer and longer T, we cannot simply use long tau values because at some point we are limited by relaxation. So this is one very important point. And also in terms of sensitivity, you could think, okay, maybe I don't need this maximum resolution, so I could basically record a shorter time trace and thereby use a longer tau value to get more sensitivity in my measurements. Another thing that I want to point out here is that all the PDHS time traces that I show are recorded with an RF phase cycle. And the reason that we do this is that if we apply the PDHS sequence without the radio frequency pulses, or basically keeping the RF pulses off resonant, this microwave pulse sequence is the 4-pulse ESM sequence, or high score sequence in a way. And this microwave part of the sequence already leads to a background signal that you can see here in blue. And to get rid of this background decay, we can now apply a simple RF phase cycle to basically get only the modulated part, and then we basically obtain PDHS time traces without any background signal. Okay, so I now want to talk how can we extract distance distributions from PDHS. And for this, I want to quickly remind you of the signal that we detect for each distance. So if we follow the product operator formalism for the PDHS sequence, we realize that because we use the preparation block in the same way that we also use it for frequency domain ENDOR, we are still dependent on our blind spot function. And this means that our ENDOR spectrum will basically be the classical Peak pattern as a product with the blind spot function. So what we detect in PDHS and also ENDOR is this kind of MIMS Peak pattern. And if we now want to extract distance distributions, we can use a very similar approach to what people have done in electron-electron distance measurements. So what we actually want to do is we now need a time trace for every distance r that we compute and generate a so-called dipolar kernel. And because in PDHS there are two main differences that we have to remind ourselves. So the first one is that we have the effect of the blind spot function, which means that our dipolar kernel is also dependent on the experimental tau value. And another difference is that we do not have any intermolecular background signal. And the question at hand now is that we, of course, if we want to extract a distance distribution, we now want to find the distance distribution that we can multiply with our dipolar kernel to get our measured data ST. And this problem is ill-posed because there are several solutions for distance distributions that can represent our measured data. So there are two main approaches that you can use in this case. One would be to use a parametric model for your distance distribution, for example, a Gaussian. So in this case, you would basically use a Gaussian model for P and then fit your experimental data by using least square fitting. In this case, with a simple mono-Gaussian model, you only have two fitting parameters, the mean distance and the width. And as soon as you increase to bimodal, you also get for example, a weight for each Gaussian and so on. However, you can also use an approach without any prior information to your distance distribution. And this would be to use, for example, methods like Tikhonov regularization. In this case, P is basically, the distance distribution is basically a vector, and every element of the vector is a variable parameter, so a histogram. To now find the solution, we introduce this regularization parameter lambda that basically scales controls the smoothness of our distance distribution. And this means that we basically say, okay, our distance distribution has to fulfill a certain smoothness to be a valid solution for our problem. We can now use these two approaches to simulate our data. So if we now look at the time trace, we can get a nice fit using a simple Gaussian model, because for these model compounds, we only expect roughly one distance of the fluorine nucleus, because we have kind of a symmetric linker. And you can see that we can get a nice fit for both time domain and frequency domain. And the distance distribution that we get from our fit also shows a similar distance to what we expect from DFT optimized structures of this model compound. We can also take the second approach and perform Tikhonov regularization. In this case, we need to find a nice way to choose our regularization parameter, and one criterion would be to use the L-curve criterion, where you sit at this maximum point of curvature here. And in this case, we also get a nice fit of our data and a similar distance distribution for our fit. Okay, so I've now shown you some nice results for this 15 angstrom model compound. However, we wanted to also look at the limits of our method, and this basically led us to to synthesize new nitroxide model compounds with even longer fluorine distances, 18 and almost 20 angstrom. And also we wanted to look at the effect or how reliable PDHS can extract distance distributions. And for this, we looked at this model compound where we have 4 flexible bonds that will, or like 4 bonds that will influence the distance distribution of our fluorine nucleus to the nitroxide. So let's first look at the kind of ruler molecules, which have very rigid linkers. We are able to nicely obtain PDHS time traces for all of them. And even at a distance of 20 angstrom, we are still able to nicely resolve the principal shape of the Pake pattern. And this is really great because it's still one of the first examples where such a small hyperfine coupling has been measured using ENDOR. And we can now use the approach of using a Gaussian model to fit our data. And in this case, we end up with these 3 distance distributions for the different model compounds. And you can see that for all of them, we have a reasonable agreement with the DFT output. And what's also interesting is that we see a similar effect that is known from DEER, that by increasing the distance, we obtain wider and wider distance distributions. And this is, of course, makes sense because basically by increasing the linker length, we have more degrees of freedom and larger flexibility. So one important point that I want to talk about now is what is actually the maximum limit of PDHS. So how far can we actually go? Because you can see at 20 angstrom, we can still nicely resolve this coupling. And for this, we basically first did some calculations. So we plotted the experimental values and then approximated basically by looking at the line width that we obtained for our rigid linker molecules, what could be the maximum distance that we can achieve. And you see that we get kind of a distance limit of around 30 angstrom. However, I just want to point out again that in these cases, for 30 angstrom, we really have to consider sensitivity, because to at least record only one modulation period in PDHS, we already need tau RF values of 360 microseconds. So if we want to see any kind of distance distribution, we would need even longer tau RF values. And again, in this case, the blind spot function really is limiting, and this sensitivity relation is really kind of a problem here. So it will be interesting if we can really reach this maximum theoretical distance limit. Okay. After we have looked at the rigid compounds, let's look at the flexible model compound. In this case, with frequency domain ENDOR first, again using long RF pulses to have maximum resolution, we see that we only resolve one single splitting of 72 kilohertz. And in PDHS, we see a difference now. We see that we actually resolve 2 splittings, 64 and 128 kilohertz. And if we now apply again our model and use a bimodal Gaussian to fit this data, we can get a nice fit for our time trace and for our spectrum. However, for frequency domain ENDOR, we could simply use a mono Gaussian model and some kind of ENDOR line width. And the reason here is, of course, that the resolution in frequency domain ENDOR basically is not sufficient to separate the blue and the red case here. So we cannot. We could also simulate the frequency domain ENDOR spectrum using the blue distribution. However, there's simply not enough information in this spectrum. However, to kind of now validate that the distribution of PDHS is correct, we also performed DFT calculations. And here you see basically the ensemble of the 21 most relevant conformations. that we found for this model compound, and you see that for some of the conformations, the fluorine is pointing at a closer average distance of around 10.2 angstrom, and for some conformations a little bit further away to around 13.2. And if we now basically take each of these conformers and generate a distance distribution, we see that our DFT model shows very close, is in very good agreement with the distance distribution that we have, that we extracted from our PDHS data. Okay, in the last few minutes of this talk, I want to shortly talk about multispin effects in time domain ENDOR. And this is work that I have been doing closely with my colleague Sergey. And we have looked now not only at molecules that contain a single fluorine nucleus, but molecules that contain 2 or even more fluorine nuclei. And there's a fundamental difference now for these molecules, because if we look again at the preparation part of our sequence, so basically the MIMS block, we now, in the case of 2 fluorine nuclei, we not only generate electron nuclear 2 spin order that we know for from the single fluorine case, so 2 spin order for nucleus 1, 2 spin order for nucleus 2, but we also generate this 3 spin order term, SZ, IZ1, IZ2. And this term will also manifest itself in the time domain ENDOR experiment. So if we now move on and think about what we will detect here, we can look at the nuclear sublevels, and the electron nuclear 2 spin order terms will generate us nuclear coherences that will lead to modulations with cosine hyperfine coupling T, so basically single quantum transitions in the nuclear sublevels. And the 3 spin order term will generate 2 spin nuclear coherence. And in this case, we detect the sum and difference of the hyperfine couplings, which you can think of as the double quantum and 0 quantum transitions in these in the nuclear sublevels. And there's actually, in this case, this was already proposed in the initial analysis by Gempale and Schweiger. But it was still kind of tough to measure these effects, and we have now looked at this model compound here, and by using clever RF phase cycling, we can basically isolate these 2 components. So if we use the black phase cycle here, which is the same that we also used for the single fluorine case to get rid of the background signal, we get the PDHS signal that that we all know by now. However, if we use a phase cycle that is basically a double quantum filter from NMR, we see that we can obtain a PDHS time trace with a modulation with twice the hyperfine coupling, because in this case, the hyperfine couplings of both fluorines are very similar. So we basically obtain 2 times the hyperfine coupling. And you can see in the frequency domain that we now have the Peg pattern with twice the splitting that we have seen before. And this, of course, could be very interesting because we can basically correlate hyperfine couplings and also maybe extend the resolution limit of PDHS even further. One last point that I want to talk about is that when we initially started these experiments using this this compound, I was setting up the experiment and I used a tau RF value of 420 microseconds, which was kind of the standard that I used for the other model compounds. And I looked at the time trace, and you basically see nothing. And I was very puzzled because RF pulses were set up correctly, microwave pulses were set up correctly. So what's happening here? And if I switched the tau RF value back to a shorter time of 220 microseconds, I was able to record the time traces that you see here. And this actually now made us curious. So we thought, okay, what's happening here? So we recorded the nuclear spin echo as a function of the delay time tau RF. And what we saw in this case is that we have a strong modulation of the echo envelope, of the nuclear echo envelope. And at a time delay of 420 microseconds, we actually have no nuclear echo signal. And this, if you think about it, is of course makes sense because in multifluorinated systems, we not only have heteronuclear dipolar coupling, but we now also have homonuclear dipolar coupling, which is not refocused in the nuclear spin echo. And if we now calculate the theoretical dipolar coupling for the distance that we can obtain from DFT, we see that we obtain a coupling of 1 kilohertz, which is exactly the same coupling that we also see in our experiments. So with this method, we should be able to actually measure nuclear distance distributions via EPR. Okay, so I again want to conclude and kind of highlight the key takeaways and also look at future perspectives. So I have shown you that for nitroxide model systems, dead time-free time domain ENDOR shows increased sensitivity when compared at equal resolution, and that PDHS significantly enhances the resolution and thereby pushes the distance limit, but also allows us to extract reliable distance distributions. The next step now, of course, would be to step away from the model systems and now apply these new methods to relevant biological systems like RNA or proteins. And of course, here there are new challenges because we have lower concentrations and more challenging relaxation properties. However, initial experiments with RNA already kind of work out. Another point that I've shown you in the last few slides of the talk were multi-spin effects in time domain ENDOR, and we also want to further explore these interactions and maybe use them to detect nuclear distance distributions and also push the resolution even further. So with this, I come to the most important point. So I want to thank all the people that made this work possible. So of course, the whole Benati group and especially the people who have contributed a lot, marked in red, and also my PhD course for funding. And I'm very happy to answer any open questions.
All right, thank you very much, Lucca, for this very exciting talk. These are impressive spectra. So now we open the Q&A session for the audience. So I encourage anyone who has a question to unmute themselves and ask the question directly. So while the people are thinking about questions, I have maybe a couple very naive and trivial ones. So you said at the beginning that you chose Q-band because it's of course is a widespread machine in the EPR community, and the power is enough. So it's available. You have a high power amplifier. So my question is, do you see any advantage or disadvantage going to high field, and what are the limitations or the gains there?
So I think good question. I think in principle, the RF power is really one of the most limiting factors. So I mean, at W band, for example, in our lab, it's not straightforward to use such high RF powers and not burn the ENDOR coil in the process. And I mean, in principle, if we are aiming for very small hyperfine couplings, I mean, we could also first try to do this with longer RF pulses. But yeah, I mean, I think we are just at the start. So there are also different ideas, for example, by coupling using, for example, kind of matching box and use this to increase the excitation profile of the RF pulses. But yeah, I think there is a lot of room for new ideas, and yeah, I think we are not restricted to only low field, but we just have to find a way how we can use it at higher fields at some point. Yeah.
All right, thank you. Yuji has a question, so please.
Hi, Lucca. It's an excellent talk. Thank you very much. I have a small question. So at the end, you mentioned that because there are two fluorine nuclei, so you cannot refocus them. So have you considered using some homonuclear recoupling, refocus sequences from NMR?
So yeah, we thought about it. I think yeah. So these results are very fresh, and we are just starting to understand all the effects that we see with multiple fluorine nuclei. But yeah, I think this is the really nice thing about this whole time domain ENDOR approach, that we really can take a lot of ideas that are known in NMR from very long time ago, like 60s or something. And we can now really use all these tools to play around, because we now have the freedom by refocusing stuff in the time domain. We can now really look at different interactions isolated. So yeah, I think could be an idea to also refocus these interactions. So yeah, in principle, I mean, adapting these ideas from NMR is exactly what we kind of started to do now. But yeah, so would be an option.
It's really nice. And another small question is when you showed the result for the different concentrations. Sorry for the noise. So I see for the background, you see more of the ripples instead of like a pure noise from the frequency domain ENDOR. So do you know like where it may come from? Yeah, here.
You mean this?
Yeah.
Yeah. So in principle, I think this is kind of a difficult question, how to really fairly compare the signal to noise ratio, for example, of frequency domain and like Fourier transformed spectra, because you do Fourier transform in the process. So the noise level depends a lot on how you perform Fourier transform in the process. And so yeah, it slightly also alters sometimes even like the side shapes and like these wiggles. But yeah, I cannot really tell you where exactly now there's like what kind of noise leads to these ripples. But in general, they increase, and it's also dependent how long your zero filling is and stuff like this. So yeah, I think it's kind of also a way how you process the data. So this was in general, I mean, the 2 micromolar measurement was very tough, and it was more a proof of concept that we can go for these very low concentrations and still get at least some ENDOR spectrum.
And I don't know whether I have time for another small question. I will just
think you
ask.
can go ahead. Just go ahead. Yeah.
Right. So when you showed the PD hyperfan spectroscopy, so you have another pi pulse in the RF, in the microwave here. So is it very sensitive to the B1 inhomogeneity, or you don't see too much problem?
So I mean,
does it need to be a perfect pi pulse over the whole sample volume?
No, I think it's more like, I mean, it's basically kind of a pump pulse. So we just apply maximum power and try to hit as much of the EPR spectrum that we can do. But yeah, we have not seen any problems with B1 inhomogeneity so far. Yeah, I think but to optimize every single part, could, like, we have not looked into everything in close detail. So yeah, maybe there's still ways of improving.
Okay, Thank you so much.
All right, we have a question from Julian. Please go ahead.
Yes, hi Lucas. Lucca, very nice talk. Also nice project. I want to ask for slide 15, where you were before, the time traces there. How does this relate to A? Like it starts at the center of the echo and then goes down, or like how does the time trace relate to the, yes?
So you mean, yeah. So we use a very short tau RF here. So it's basically only 2 microseconds to basically have to keep this T as short as possible. We basically only the nuclear spin echo is almost inside the RF pulse basically. And but it's still enough to basically then step this RF pulse over these first few points. And I mean, it gets better with increasing hyperfine because there the dead time is not that important anymore. But yeah, so in principle, we also did experiments where we used longer tau RF. So we really first just detected the echo itself. So yeah, but it's a very good approximation that basically the echo comes after the center, like it's written here basically. So it's a good approximation, and then you can cut off the first few points of what you detect, and then you are at the start. Yeah.
And it's symmetric?
In our cases, yes.
Okay. And if I may, I have one more question.
Of course.
You mentioned that like it's difficult to adjust your sequence because if you want to go to very like long distances, you have the relaxation time and so on influencing everything. What parameters can you change to like tweak your experiment in the right direction? Did you measure at different temperatures as well, or is this not useful?
Yeah, I think this is useful. So I think this is like exactly what I wanted to kind of achieve with this slide. So yeah, I think it's really dependent on what you are aiming to do. So if you are going for maximum resolution, maximum sensitivity, you really have to look basically at this, at the influence of your phase memory time to choose your tau, and you also have to have sufficient long T1. So you kind of need to change temperature dependent on T1. But yeah, of course, it's I mean, I focus now mainly on nitroxides, but I think these sequences are not limited to any kind of spin label or nucleus, so they could be applied to different systems. And so yeah, I think most importantly, it's like you should really be careful with your choice of tau, and but also yeah, to at least have sufficient long T1 to of course fit the sequence in. So for us with nitroxides, it seemed 50 Kelvin or below seemed good. A good also yeah, good agreement between like short repetition time and long T1. Yeah.
Okay, but you did not measure other temperatures yet, or?
Yeah, I don't have them here, but we also, in principle, it also works at lower temperatures and even higher. For us, I mean, we used same parameters so far, so we really didn't really optimize everything there. So, but yeah.
Okay. Good. Thank you.
All right. Any other questions from the audience? Doesn't look like. So I think we can close the session here. So thanks, Lucca, again for this very nice talk. I'm looking forward to seeing more of this in the literature very soon. I mean, it's very impressive results. So thank you everyone for joining the seminar, and yeah, I'll see you in a few weeks for the next one.
Yeah.
Bye.
Thanks.